Okay, welcome everyone. We're at the top of the hour. We're going to go ahead and get started. My name is Cheryl Rogers, and I am the Director of Marketing at Golden Helix. Our webcast today is presented by Greta Lindsay Peterson, our Director of Product Management and Quality. Greta is going to discuss using mixed models to adjust for population structure in a rare variant burden test. Greta, I'll allow you the honors.
Thanks, Cheryl. All right. So today, in the abstract, we had talked, or I had mentioned that I would be able to compare our method that we've developed to other methods. And I just want to say right off the bat, unfortunately, that didn't work out, and I wasn't able to get the other R functions working in a timely manner to compare against, to do a complete one-to-one comparison with our models. So if you're looking forward to that, you're not going to see it today, but we will be presenting that content in the future. Today we will compare this new method with our existing KBAC method in SVS. So with that, let's get started. Just a second. There we go. All right, if you have any questions during the presentation, please feel free to enter them into the questions pane in the GoToMeeting interface at any time. Due to the technical nature of today's content, if I do not feel like I can answer a question in the detail required in the time allotted, we will get back to you via email or phone as required. First, let's just, for those of you who might not be familiar with Golden Helix, we have a flagship software product, SNP Variation Suite. And SVS is a solution for data management, research, and analysis of genetic data, including microarray genotypes, or GWAS, DNA-seq variant calls, and copy number log ratios. We also have a free genome browser, aptly named Golden Helix Genome Browse, and we've now included that into SVS for enhanced visualization of association test results in conjunction with annotation sources. Golden Helix also offers services, some of which are listed here. If you need assistance or would like somebody else to do the analysis for you, please contact us and we can see if that's something that we can help you with. We also have support listed here. We are known for our legendary support, and if you had not had a chance to experience it, I would encourage you to try a demo of SVS and also try out our support. I am the manager of our customer support team, so I, of course, very proud of it. And more specifically, the methods that we have developed are for our SVS program. So let's give a little orientation about SVS. We created this program specifically for genetic research, and it was initially developed in the GWAS era. The performance, usability, and visualization have made the software an obvious and popular choice for genetic researchers, and has resulted in hundreds of citations and peer-reviewed articles. More recently, as the world of genetic research evolves, the software has added numerous tools to account for a variety of workflows. SVS now includes GWAS, CNV, PBAT, RNA, and DNA sequencing methods, as well as an incredible and intuitive visualization powered by Genome Browse. Again, Genome Browse is completely free, so you can feel free to download it and try it at any time. Typically, putting on my project manager hat now, typically only implement published and widely accepted methods, such as KBAC. Today, however, I am going to talk about a method that has not been published yet, but we feel is a logical extension of existing methods, namely mixed models and KBAC. The way that we decided to implement this, just a little bit of a history, we researchers in the early days of GWAS analysis looked at different ways of performing association tests for complex diseases. And so we had the basic GWAS. However, limitations were discovered, in including these study designs were not able to aptly or correct for population structure, batch effects, and genotype calling biases. There's still too many false positives. From here, research split into at least two camps. Technological improvements made large-scale DNA sequencing possible on large cohorts, and to look for associations between rare variants and complex diseases. One such method for analyzing the burden of rare variants is KBAC. Improvements in mixed model algorithms and improvements to computers themselves made accounting for population structure and GWAS analysis possible via mixed model methods. However, today we are going to talk about bringing these two approaches back together into a method we're calling MM-KBAC, or mixed model KBAC algorithm. Let's talk a little bit about the study design for this method. For rare variant analysis in general, there are two different main types of study designs. The first is what we call large N, or a large cohort population, and you have a large number of cases. Ideally, you'd have matched controls or a quantitative phenotype for your complex trait. Then we also have what's called small n, small nuclear families or an extended family with a handful of members. This design is looking for highly penetrant variants for a heritable trait, either de novo mutations, recessive, or compound heterozygous variants. But that is not what we're going to be talking about today. We're going to be talking about large N study designs. And the assumptions for a large N study design are fairly rigid, and if followed to the letter, require performing analysis for all populations or subgroups separately, and then performing a meta-analysis to bring the results back together. But how often do you have a perfect study design? Not very often, I'm guessing. So what if we have violations of the assumptions, and in thousands of samples, who wouldn't have violations of these assumptions? In particular, what if you know there is some population structure and you cannot guarantee independence between your samples? Or you had to borrow controls, such as 1,000 genome samples, or you want to include multiple families with either affected offspring that all exhibit the same phenotype, or multiple large extended pedigrees of unknown structure. If only there was a way that we could account for this in a regression model. Just add random effects. At ASHG last year, I was attending talks addressing this very topic and realized, of course, mixed models with a kinship random effects matrix is perfect for this problem. Just last year, we implemented MX and SVS, which uses a linear regression model and includes a random effects matrix, a kinship matrix. So I figured it would be trivial to add a random effects component to the collapsing methods we have in SVS, which have regression versions. In the heat of the moment, I forgot that while one of our collapsing methods, CMC, can use a linear regression model for a quantitative trait, the rare variant only collapsing method, KBAC, requires a binary case control trait and uses logistic regression. Okay, well, MX can take a binary trait, and linear regression is justified when there are roughly the same number of cases and controls, and then it behaves like an Armitage trend test. So could we do that, or, well, should we do mixed model logistic regression? Everything that I heard was that mixed model logistic regression is hard, and although theoretically possible. When I talked to our senior mathematician and software engineer, James Grover, he said that KBAC really needs a logistic regression model. And he went and investigated whether he could add a random effects matrix into logistic regression. What is going to follow today is his work, and I'm very pleased to present his methods today. I will use some pictures to describe the problem for those that are not familiar with rare variant collapsing methods, but after that I will be presenting the algorithms with the formulas used. So if matrix algebra is a little bit frightening to you, don't worry. After we work through the details of the method, I will present some preliminary results and comparisons, again, comparisons between KBAC without the random effects component and KBAC with the random effects component. But first, let's talk a little bit about the data set that I used today. I did use the 1,000 genomes pilot 3 exons data set with the simulated phenotype. And this is an older data set, but it's a nice clean data set, exons primarily only. We did have to do some filtering to remove any variants that were outside of exon regions. And there are newer 1,000 genomes data sets. This is one that we have prepared and ready for our KBAC tutorial, or variant analysis tutorial. So I felt that it was good to use that in this case for this demonstration purposes. I did modify the phenotype a little bit from the tutorial data set, but you can see that we have, here's the plot of the case status for the populations. And then back to the PCA plot, you can see that there is quite a bit of population stratification here. There's 7 populations, and this is the standard 3-axis PCA plot that we normally see. And for the case frequency, we have a higher frequency of cases in the YRI population and the LWK population, as well as CEU. But the proportion of cases and cases to controls is actually quite low. We only have a total of 48 cases compared to 649 controls in this particular case. So this is not a balanced study design at all. And we're already violating both the balanced design and the population, single population assumptions. All right, other than populations, are the samples related? This is an identity by state heat map, and you can see that there is a lot of structure in this data set, again within populations, but also some of these samples are highly related to a lot of the other samples. And you could remove those from the analysis. And today we're just going to keep everything in, and we are going to account for this relatedness in our random effects matrix. Okay, mixed models make sense, but why KBAC? Of the collapsing methods we currently have implemented in SVS, CMC and KBAC, KBAC is the rare variant weighted burden test, while CMC adjusts are both common and rare variants. KBAC also uses a score test, which should be trivial to calculate once we have computed the reduced model. In the same manner that I'm going to demonstrate today, mixed models or random effects matrix can be added to other regression-based burden and kernel tests using the same principles. Again, adding it to linear regression models is trivial, has been solved. We've are presenting the solution to the logistic regression model. And now, with both logistic regression solved and linear regression solved, we should be able to add this to anything. So what is KBAC? KBAC stands for Kernel-Based Adaptive Clustering and was published in the Liu and Liao 2010 paper shown here. This method catalogs and counts multiple marker genotypes based on variant data. It assumes that the data has been filtered to only rare high-quality variants, and ideally there are at least 3 variants in a region. It performs a special case control test based on the counts of variants per region, in other words, a gene or a transcript, and the test is weighted based on how often each genotype is expected to occur according to the null hypothesis. Genotypes with higher sample risks are given higher weights. It's primarily a one-sided test, which means it is designed to detect higher sample risks associated with case status. If we want to look for protective genes, you can just invert the case status and run the same one-sided test again. All right, so let's use some cartoons to help us understand this method a little bit more. So here we have a set of controls and a set of cases, and we can imagine that these 5 controls and 5 cases are actually a lot more than that. And the light boxes are the variants, and the blue boxes we can consider to be reference alleles. We have 2 genes, and we have 2 known SNPs. KBAC requires pre-filtering to remove common variants, and we can also consider known SNPs to be common, so the next few cartoons will demonstrate that filtering process. So the first step is to remove common and or known SNPs. So you can see that these known SNPs we've highlighted in red, dotted lines, and we're going to remove those from our pool of variants. And after we remove those, you can see that there are no other variants that are at high frequencies that warrant removal for this condition. The next step is to remove variants outside of genes. Now technically KBAC will take the implementation in SVS takes a gene annotation source and decide which variants to keep and how to combine variants into the multi-marker genotype. So we don't technically need to remove the variants that are outside of gene regions, but you can pre-filter to remove those or just do it all at once. But we have highlighted these just to indicate that they will not be included in the KBAC computation. So what is left are the rare within gene variants. And KBAC essentially compares the variant count between cases and controls for each gene. So genes that have a larger number of variants and cases compared to controls will result in a higher KBAC score. So in this case, this gene on the left will have a higher KBAC score than the gene on the right. And you can see that there's quite a bit more variants in cases compared to the controls for this particular gene. So here's the specific one-sided KBAC statistic formula. The KBAC statistic compares the difference of weighted multi-site genotype frequencies between cases and controls. Here the A stands for affected and the U stands for unaffected. We can get a two-sided test by squaring the one-sided statistic, but this is not a true two-sided test in that it's always going to favor sample risks compared to protective effects. In this method, adaptive weighting is used for each genotype according to the area of the curve from 0 to I of the known component of the distribution, where I is the estimated sample risk for genotype G. This known component is called the kernel, and that's the K part of the KBAC statistic. And there are three methods that exist for computing these weights. The first method is the hypergeometric kernel. And this is the exact form of the kernel. Under the null hypothesis, conditioning on the genotype counts and the count of cases, the number of diseased individuals or cases having the multi-site genotype follows the hypergeometric kernel. The marginal binomial kernel, if the proportion of cases to controls is small and the number of variant genotypes for cases is also small, the number of cases having the multi-site genotype approximates a binomial distribution. And finally, the asymptotic normal kernel, for large sample sizes, at least 400 cases and controls, we can use the fact that under the null distribution, the sample risk for the multi-site genotype is asymptotically normal. All right, so we have our statistic, and now we need to determine a p-value to compare our results. Since the distribution of the p-value is unknown, permutation testing or something approximating it must be used to determine a p-value. For large sample sizes, a Monte Carlo approximation method for finding a p-value has been developed. As variants are rare, the number of cases for each multi-site genotype approximates a binomial distribution. Due to the low frequencies of each multi-site genotype containing rare variants, we take these counts to be independent of each other. And the Monte Carlo simulation algorithm is as follows. So we simulate a k vector of independent binomials, where k is the number of permutations, and then we compute a KBAC statistic based on the k vector using each random count as the number of cases for the multi-site genotype, pulled from the binomial distribution. Then we repeat these steps for each multi-site genotype. And the p-value is determined by taking the count of how many KBAC statistics are greater than or equal to the original statistic divided by the total number of permutations. Now in SVS, we count the original statistic as one of the permuted values, statistics computed via permutation. All right, so that's KBAC. Now let's talk about the logistic mixed model equation. The mixed model regression score, KBAC score, is similar to the KBAC regression score. We've changed the terminology a little bit to match with our existing mixed model terminology and formulas. And the component in red is what we've added with the random effects matrix. But suppose we have L covariates, such as age, sex, or eigenvectors for genotypes. Then we define the XFJL to be the value of the Lth fixed effect covariate for the jth sample. And also suppose we have random effects uj. Then logistic mixed model equation is shown here. Now this component is the value that we're iterating over. In this case, it will be each gene, the multi-site genotypes. And again, we have the score statistic. All of this is the same except for down here, again, we have the random effects component for the jth sample. Under the null hypothesis, the xi do not contribute to the solution of the original model. Thus, beta naught and beta FL and the predictions of uj are also under the null, the solution of the reduced mixed model using the same covariance matrix G for the uj as before. The reduced model can be rewritten in this expectation form, where E of y, the expectation of y given u can be expressed in this manner, and the variance of y given u is equal to A, where A is the variance of the binomial distribution itself, with the diagonal elements equaling mu j times 1 minus mu j, again on the diagonal, and off-diagonal elements are 0. The linear predictor for this model is eta equals x beta plus u, and h is the inverse link function for the model. To solve the logistic mixed model, we iterate back and forth between creating a linear pseudo-model and solving for the pseudo-model's coefficients. To determine how to create the pseudo-model, we start with a first-order Taylor series of mu about some given tentative values of beta and u, namely tilde beta and tilde u, where the tentative value of delta is a diagonal matrix of derivatives of the conditional mean evaluated at the tentative values of beta and u. And a little bit of rearranging leads to this long formula, which the expected, the left side of this is the expected value conditional on u of p. And the variance of p given u can be expressed in this form. And the off-diagonal elements of the A matrix are computed using the tentative values of mu, and the off-diagonal elements are still 0. So again, to compute the logistic mixed model, we're going to iterate back and forth to calculate those tentative values. And as an alternative, rather than using the prediction BLUP of u from the pseudo-model, we can use the expected value of u, which is 0. Now, we want to use Emma to help us solve this model and make it faster and more feasible on a genome-wide scale. But Emma assumes that the variance of the errors is a multiple of the identity matrix, which is not what we have here. Thus, to use Emma, we must first transform our pseudo-model to another model. So we want to find a matrix T such that the variance of T epsilon is equal to the identity matrix. And then we can use, we can rewrite the formula here, and we can use Emma to solve the mixed model expressed in this manner. The variance of epsilon star is proportional to the identity matrix, and it can be shown that this is solved by letting T equal the square root of the tentative A matrix times the diagonal matrix of derivatives. All right, let's summarize the algorithm. So first we want to pick starting values of beta and u, such as all zeros. Then we can repeat the following steps until the changes in beta, tentative values of beta, and tentative values of u are sufficiently small. So we can find eta tilde and mu tilde from the original linear prediction equation and the definition of H. Then we can find the diagonal tentative delta matrix, find the pseudo-model P, find the diagonal matrix T, and then solve the following for the new values of beta and u using EMMA. And again, the alternative method modifies step 5 to use EMMA to determine the variance components and to find new values of beta while leaving the value of u at its expected value of 0. After convergence, this alternative method predicts the value of u and computes the final values of eta and mu from this prediction. So that's simple, right? Well, we have implemented it in SVS and we do have results. But before we get there, let's talk about how we need to prepare our data to run this method. So we start with high quality variance. And it should be noted that there are 3 components that you need for this algorithm besides in addition to your high quality variance. So first, you obviously need a case control phenotype. And second, you need a kinship matrix that is either computed from the data or derived from a pedigree file. You can also have covariates such as age, gender, or eigenvalues from the data. And you also need your rare variant genotypes. If you want to compute the kinship matrix from the data, we start from the pool, and to get the rare variance, we start from a pool of high quality variants, and we split these variants into 2 groups. First, you have a group of common variants, where the alternative or minor allele frequency is greater than or equal to 0.05, and a group of rare variants, where the alternate allele frequency, or MAF, is less than 0.05. You can use a different MAF threshold if you have more samples. You could be let this value be smaller. If you have less samples, you can increase the threshold. Here we're just using our, the typical GWAS definition of if a variant is rare or not. From the common variants, then you can compute the identity by state or identity by descent kinship matrix. You could also use a genomic relationship matrix, some sort of pairwise comparison between samples to indicate the relatedness. If you use a pedigree to explicitly specify the kinship matrix, that would be computed from the pedigree and then not from the genotype, so we do not demonstrate that here. From the pool of rare variants, you would use this pool to perform the MM-KBAC association testing, or if you want to perform KBAC without the random effects matrix, you would use that, run that on the pool of rare variants as well. And just to show how easy it is, or the interface in SVS, I just want to point out the dialogues for KBAC and mixed model KBAC. All of the options are very clearly laid out and easy to specify. When I was trying to run alternative tools in R, I was having a lot of issues with making sure that I had all of my parameters specified appropriately. And they also ran only on a single gene or set of variants at a time. In SVS, you specify your number of permutations, adaptive permutation threshold. You can compute 1 or 2-sided statistics, and then you also select your gene annotation source. And so SVS will run your analysis on all genes at the same time. So you just have to select your parameters once, hit OK, and let it run. you don't have to worry about writing wrapper functions to run this iteratively over different sets of variants. You can also add additional covariates into the KBAC model, and I will compare KBAC with additional covariates, KBAC without additional covariates, and then the mixed model KBAC today. And there's also a different way that you can handle the genotype missing values. You can either treat it as the wild type or just ignore those samples for those particular genes. The only difference between the two KBAC with regression and mixed model KBAC is that we also include the ability to specify the pre-computed kinship matrix, and we do have one additional option for imputing. We can say that you don't want to impute the genotype missing values, but you do want to pre-compute the reduced models, and that will be not as fast as imputing the wild type, but it will be faster than ignoring those samples. All right, so again, I ran the 3 different tests, and this is a screenshot from our Golden Helix genome browse. On the top, I have the KBAC method. In the middle is KBAC with population covariates, and you have to specify binary or numeric covariates at this time. And so I, with the 7 populations, I specified the first 6 alphabetically. So the Y or I population was left out of the model, and in the standard leave one out approach. And then at the bottom is MM-KBAC or mixed model KBAC. We also have a feature list, and I have this feature list sorted in descending order on the negative log 10 p-value. So we can see that the top gene here is PSRC1, and we can jump to that, and we'll look at that in more detail later. And the next one is the HIRA gene. And you can see that there are differences between the 3 methods. The genes that are significant are still popping up, the top gene, which is that PSRC1. But each method brings more genes to significance. And we'll look at some QQ plots to see, the effect of all of these genes. The other thing that I want to note is that, again, because there's permutation used to compute the p-values, the p-values, most significant p-values will only be as significant as the number of permutations that are allowed. And for this test, I only performed 10,000 permutations. And so the largest p-value possible is a value of approximately 4. To get more significant p-values for your significant genes, you could allow for 20,000, 100,000, 50,000 permutations. It'll take longer to run, but your top results will be more significant. So for the QQ plots, I just compared KBAC and MM-KBAC. And normally in a GWAS study, we would see inflation in a QQ plot where we do not have any correction for population. In this case, we actually see the opposite, and our pseudo-lambda value is quite a bit less than 1. When we look at the QQ plot for MM-KBAC, we can see that it is more what we expect. The values follow the y equals x line for quite a bit, and our pseudo-lambda value is a very nice, slightly over 1 value. KBAC with population covariates, it's still less than 1, but it is better than not including the population covariates. So let's look at those top 2 signals again. Again, these are images from Genome Browse, and I'm also showing the Cosmic Gene Database and OMIM genes, and I am displaying the negative log 10 p-values in the plots, and I hope that you can see those. And down below are all the variants from the genotype file, and then we have the gene track at the bottom. So we can see all the locations of all the variants that are included in this statistic. And we can also see that all these values are roughly the same, but KBAC and MM-KBAC are the same compared to KBAC with population covariates is a little bit smaller value. So this gene could warrant further examination if this was a real phenotype. Again, it's not in known databases, so it might not be that interesting. The next gene that we'll look at, HIRA, it's not as significant, but with more permutation testings, perhaps it might be. It is a gene that is in the OMIM gene database. It's also in Cosmic, and MMKBAC has the highest p-value out of the 3 methods, and you can see the variants that are going into this gene. If we were in Genome Browse, we could scroll in and we can see these variants in more detail. I also want to note that I have these variants grouped so that the controls are at the top and the cases are at the bottom. Again, because we have such a small number of cases to controls, there's just a small bar at the bottom. So this method, we're looking forward to add into SVS in the near future. Right now, we're just working on optimizing the algorithm to make it run faster. And we're also, right now, it can use, the permutation testing can take advantage of multiple cores on Windows machines, but we're going to make that work as well on Mac and Linux computers. And we're running this on GA17 data and 1,000 genomes data. But if you have real data and are excited to try this out while we are optimizing the algorithm, let us know. We would be happy to add you into our early access program. And we have submitted an abstract to ASHG for this method, and at that time, hopefully we'll be accepted. We will have more results comparing this method to alternative methods such as famSKAT, VC score, and any other family-based collapsing methods that might be out there at that time. I just need a little bit more time to figure out how to get those R programs to work. Now, I'll go ahead and turn it back over to Cheryl for some announcements, and after that, we'll get to your questions.
Thank you, Greta. I just have a couple of announcements. Our webcast recording and slides will be up on our website tomorrow as normal. If you haven't heard yet, we are hosting a t-shirt contest. I know a lot of you out there know and love our t-shirts, and we thought it was time for some new designs. I know that many of you have some great ideas, so I hope to hear them soon. You can find the details on our website. Right on our homepage under the Events tab, you'll see a link. You can read all of the details there, and I look forward to getting your submissions. If you're not an artist, not to worry. All you have to do is describe your t-shirt idea in writing, and we can put it together for you. Our next webcast is coming up on July 22nd, and we will keep you in the loop as we finalize the details. That's all the announcements I have, so we're going to turn it back to Greta for some questions.
Thanks, Cheryl. So we just have a few questions today, and again, if you're interested in the details of the methods, once we ship this in our software, we will have this thoroughly documented with all the formulas written out in our manual, but we can provide early access to that as well. And the content of this slide of this presentation will be available, and so you will get the formulas that we have in this presentation early as well when in the slide deck. And so we have a question, what file format does KBAC take? So KBAC, again, runs in our SVS software. So SVS can import data in numerous file formats. Basically, after importing it into SVS, you end up with a matrix of genotypes. And then in genotype, in this case, which I discovered is not the same as in R, it's the actual alleles separated with an underscore delimiter. And then we join that to the phenotype for the same samples. So you can visually see the phenotype connected with your genotypes and any covariates that you have. So again, any kind of file format that you can get into SVS, KBAC can take. So we have a question, why are we using KBAC instead of SKAT? Well, we had KBAC implemented in our software. James Grover, our developer who is implementing this method, his next project will be implementing SKAT. Once we have SKAT implemented in its SKAT-0 form, then again we can add this mixed model component into SKAT, and we would have mixed model SKAT. We wanted to start with taking the two components, so we already had the Emma-X, or Emma, and the KBAC, and bring those together, and then that would be our proof of concept, and from there we could go any number of ways. Does MMKBAC give the odds ratio or beta for each SNP? Our Emma-X implementation can give the beta and the beta standard errors, and so we would be able to bring those in to MMKBAC as well. And in the same way, the logistic regression component should be able to we should be able to bring that back into Emma-X, and we would have Emma-X be able to actually perform a logistic regression rather than just the linear regression approximation. Okay. How do you get the high quality variance at the beginning? Any recommended software for the variant detection? SVS starts after the variants are called, and the VCF files are then imported in, and to get the high quality variants, we generally filter down to gene regions, exon regions, if it's supposed to be an exon capture kit. We filter out known variants based on dbSNP. You can filter poor call rates. We have a few tutorials available on our website that talk about how you can actually get down to those high quality variants. And does permutation testing work if the covariates are correlated with phenotypes or correlated with the genotypes? So the permutation testing, we don't permute the covariates. The covariates are fixed to the genotypes. We only permute the case control status. And so the permutation does work if the covariates are correlated with or associated with the genotypes. We have not yet compared MMKBAC with Emma-X. Emma-X assumes that you are working with common genotypes, common alleles. MMKBAC assumes that you're working with rare alleles, so their assumptions are completely different. However, you could run a single SNP test on rare variant data, but the reason that collapsing methods were designed was because that the traditional GWAS methods, including Emma-X, are not designed to handle rare variants. And let's see here. Can you use KBAC or MMKBAC in plant genetics? You can use these collapsing methods. For any genome, all you need are the VCF files and a gene annotation source. So as long as you've got some sort of gene source, definitely you can use it for any kind of species. Again, as long as you have the sequencing data for that species and the gene. And let's see, we will, we had somebody ask again about SKAT. We have not implemented SKAT in SVS yet. That is going to be our next project. And once we have SKAT implemented in SVS, we're going to implement the SKAT-0 version. Then we can add this new approach into SKAT-0 to have a mixed model SKAT. So first we're going to start with implementing SKAT-0, and then we will bring this component into SKAT. All right. I think that's all the questions that we have. If we didn't get to your questions today, please let us know and email support@goldenhelix.com. Again, please feel free to check out the t-shirt design contest, and I hope to see you guys at ASHG, and I'd be happy to talk about this method more at that time. Thank you.
Thanks again, Greta, and thanks everyone out there for joining us today. We hope that you enjoyed the webcast. Like I said, I will have it up on the website tomorrow. I hope everyone has a great day. We'll see you next time. Bye-bye.